discuss about stem cells. Okay, so criteria for selecting media.
Ma'am,
Okay.
whatever slides you are showing here, can you share in group every time?
Yeah, no problem. So just text me what is the name of the group. So there are many groups now. So just text me the group name. I will send. Okay. In the
Yeah.
chat box you can do at the end of the class. I will see.
Okay.
Okay. So today we will discuss about criteria for selecting media. So I said like there are many types of media, and the composition of media also it differs from type of media. So each cell type has a, specific requirement. Okay, so based on that requirement we have to select the media. So the choice of cell culture media is extremely important. So that's why we have studied discuss we have already discussed histology, all these things, because every cell type has a requirement and we should look for the required media to be used when culturing such type of cells. So the choice of cell culture media is extremely important and significantly affects the success of cell culture experiments. So the selection of the media depends on type of cells to be cultured. So each cell type has a specific media. They grow well in a particular type of media. And the purpose of the culture. So why we are culturing the media? Culturing the cells. Okay, so we have lots and lots of experiments, essays, and purposes. Like for we might test drugs, or we might just study the cell biology, or we might study the cancer biology. So like that, each experiment will have an aim and vision to carry out particular experiment. So we have to know the purpose of the experiment, why we are culturing the cells, to do what. When we know that, accordingly we have to use that.
Pencil.
Please mute your microphones. So the simple example is, when you are growing, when you are culturing the cells, you can use serum media, serum containing media. But when you are treating the cells with some drugs or some compounds, and you want to see the effect of the compound on the cells, then you should not add serum. So why I told you this, minute quantities or components of which are present in the serum that interfere with the action of the drug. So most preferably serum won't be added when you are treating with some compounds. Okay, whether it is anti-cancerous or any other anti-inflammatory or any other compound. So that you should know the purpose of the experiment. Then resources available in the laboratory. So there are some high-end media, some normal media. So you should look at your lab facility. Okay, because uh, the media requires, it should be extremely sterile and stringent. Some stringent methods should be followed while doing cell cultures. So we should go with our facilities, whatever the facilities we have. We should not blindly use the media which is used in a very high-end laboratories. So we can go for some what you call substitutes. Okay, the same composition but which suits our laboratory facilities. So like that we have to select the media, and different cell types have a specific growth requirement. Therefore the most suitable media for each cell type is determined experimentally. So already there are so much research has been done on cell cultures. So already which cell types require what kind of media, this was already established. So we can just pick up from the group of media which is suitable for particular type of cell lines, and we can just check with our facilities. Or if you are doing something new, okay, we have to standardize first. We can use different types of media and we can see the growth of the cell lines, which is best media for growing this particular cells. For example, MEM media is most suitable, like Yeah, for adherent cells. When we are doing adherent cells, that is monolayer cell cultures, adherent cells means which will stick to each other. So in that type of cell lines, we can use MEM, whereas RPMI 1640 is used for suspension cells. So already this was established. So RPMI 1640 is most suitable for suspension cultures, whereas MEM is suitable for adherent cells. So this is just an example. You have many types of media. So see here. So you can see like these are the cell lines. These are the media. Second column and third column is which type of serum you are adding. For example, three T3 cells. We can use MEM or DMEM. Okay, so here you have the option MEM or DMEM. So based on the means availability. So always the supplier may not be able to supply MEM. So then you should quickly go for the DMEM. Okay, like that you will have the options, not a single type, but you can choose based on the availability. The same way, chick embryo fibroblast can be cultured in eagle's MEM, like that. So here a calf serum or fetal bovine serum, which one, horse serum, HOS means. So like that, suitable serum also was given. Okay, so we can choose from that. And always remember, whenever we are using serum media, see for a whole experiment, suppose you are doing the experiments for 1 month. Okay, you have planned for 1 month, so you should procure the same batch of serum for all your experiments. Then you will get a uniform result. Okay, because the serum quality will change from batch to batch. I think in the last class we have discussed this. So suppose like a person supplier is supplying only small quantities of serum, and you have to switch over to the another supplier. So the serum quantity may differ from batch to batch, and this will definitely affect the experiment. Therefore everything should be a single batch, and at a time we can procure all the chemicals, and then we can plan the experiments. So then balanced salt solutions. So balanced salt solutions are nothing but they are just salt solutions, okay, which have inorganic salts like sodium bicarbonate, and sometimes glucose also may present. And HIPAA buffer. So I told you, like a normal buffer like carbonate bicarbonate buffer also we can use, or else HEPIS, which is somewhat expensive, even that can be used if required. So if we are using HEPIS buffer, then that can be added to balanced salt solution. Okay, and equivalent amount of NaCl can be omitted. Okay, so that to maintain the osmolality. And this is the basis for complete media. So I told you, like, complete media is nothing but, so after this solution we are adding serum, then other requirements like glucose as an energy source, then glutamate, like that. If we add all the requirements, then finally it will become the complete media. And commercial suppliers will provide Eagle's minimal essential media and Hank's salts. So Eagle's minimal essential media with Hank's salts, or Eagle's minimal essential media with Earl's salts. So these two combinations are there, indicating which BSS formulation was used. So we can select any of the thing. It means the salts are different. Here Hank's salts and here Earl's salts. So what are the uses of this balanced salt solution? So it is a diluent. Suppose if you want to do like whatever, if you want to dilute or if you want to wash anything, you should not use water here in cell culture. We only use PBS, phosphate buffer saline, or balanced salt solutions, like that we will use. So here to dilute amino acids or vitamins, okay, we can use this balanced salt solution. And also for wash, okay, isotonic wash or dissection media, we can use this balanced salt solution for 4 hours incubation or like that. Means washing, what for washing is required to remove any debris, or some cells will float, okay, dead cells. So to remove all those things, usually we wash in frequently, okay, like after 2 days or like that. Whenever we do subculturing, we wash like that, okay. And to maintain the pH and osmotic pressure of the medium. So these are the salts which are present. So here Dalbecose phosphate buffer saline. So this Dalbecose phosphate buffer solution has two compositions, like without calcium and magnesium or with calcium and magnesium. So other than this, you have Earl's balanced salt solution, Hank's balanced salt solution, and Spinner salts. So the composition is almost same, but the concentration may differ, okay. In some cases you have some components are missing, but overall they are similar, okay. Here you can see calcium chloride, potassium chloride, like that. You have all the necessary salts, and sometimes glucose, phenol red are also, they will be added. So here this are absent completely in Dalbecose PBS, if you see here, right. So serum free media. So presence of, so what is the importance of, in the last class we have discussed the importance of serum, but sometimes serum free media is also very important. So the presence of serum in the media lead to misinterpretations in immunological studies. So immunological studies mostly include antigen antibody reactions, or how this what is the concentration of antigen or concentration of antibodies. So such kind of things we see. But whenever we have serum, so our immune system basically has, serum is one of the component of immune system, because we have complements which are serum proteins which are present in the serum. So most components of the serum are directly involved in the immunological reactions. Suppose if you have serum in the media, then already that serum might contain some antibodies or antigens which will interfere with our experiment. So therefore we may get wrong results, okay. So therefore when you are performing immunological studies, we should exclude serum, okay. So for this purpose serum free media has been developed. They are formulated to support the culture of single cell type and incorporate defined quantities of purified components. So serum free media is used for a single type culturing, and here without serum we usually know what are the components and what are the quantities. So I have used 1 mg of amino acids, 2 mg of so and so, like that we will know. But when we add serum, we know that okay, I have added 1 ml of serum, but what are present in 1 ml of serum, I don't know. So therefore when you are removing serum, usually you will know what are the components present and what are the quantities. So that will give you more clarity about the results when you obtain the results. So what are the advantages of serum free media? So it gives pure constituents, okay. So it will have the, like this is the standard medium. Here you have standard medium and selective medium. So standard medium means you know the constituents and you will know the formulation. And in the selective media, so one of the major advantage is control over growth promoting activity by serum free media, and this ability is to make a medium selective for particular cell type. So that means selective media means you are just giving something which is very particular to that cell type. Like here you have example, fibroblastic overgrowth can be inhibited by using MCDB 170 and 153. So that means If you want to inhibit the growth of some cultures, you can add some chemical. Or if you want to induce the growth of something, you can add some growth factor like vascular endothelial growth factor or epidermal growth factor. So like that, this is selective to only that particular type. So you should have a great knowledge about what type of cells, what type of experiment I am doing, what is the purpose of the experiment, and accordingly you can select the media. That is selective media. Standard media means it is not like that. So you have some types of cells for which this media is good, like DMEM or MEM, and you will use it. Some may grow well or may not, whatever the experiment, you will choose the one of the best medias which are available. Okay. Then advantages of serum free media is regulation of proliferation and differentiation. So these are some of the advantages. Like when you know that serum is absent in your media, you can definitely choose what components you want, what are the quantities you want, everything you can choose. But when serum is present, the choosing will be limited. Okay, so the disadvantages are multiplicity of media. So each cell type appears to require a different recipe, and cultures for malignant tumors may vary requirement. Okay, so that's what I'm telling, like each cell type has a different requirement. So serum always may not present a good environment for culturing. Okay, even tumor cells also, each tumor cell type requires a different type of media. Then reagent purity. So here the removal of serum also requires that the degree of purity of reagents and water, as the removal of serum also remove the protective detoxifying action, and because some serum proteins which are present in the serum, so they will give some detoxifying action. So that means the presence of serum is good in some aspects, but we don't know exactly what component is contributing to what type of action. So when you have such kind of confusion, it's better to remove the serum free. But if you are just growing the cells or you are not specific to do anything, then you can definitely add serum containing media so that it will give you some protective detoxifying action, and also the growth will be very good when compared to serum free media. Okay, so these are the disadvantages of serum free media. Then replacement of serum. So serum can be replaced with each individual components. So the essential factors in the serum have been described and include adhesion factors like fibronectin. So usually in serum we see fibronectin, which is very good for adhesion, that means attachment. So when you are doing monolayer cultures, adhesion is very important. So therefore adding serum can give you a good growth. Then peptides such as insulin, platelet derived growth factor, TGF beta, tumor growth factor, that regulate growth and differentiation. Then essential nutrients like minerals, vitamins, fatty acids, intermediary metabolites, so all these are present in the serum. And hormones like insulin, hydrocortisone, estrogen, triiodothyronine, so all these are present in the serum. So these constituents, they regulate membrane transport, differentiation, and cell surface constitution. So all these functions are mediated by the components of the serum. Some of these constituents are included in the formulation of serum free media, and others may not require. So that means when you are using serum free media, suppose, then what you can do, you can simply add whatever is required. Like suppose I want monolayer cultures, but I am not adding serum in the media, so I can just add fibronectin. I can just buy fibronectin separately and I can add that, so that the purpose is served, but other functions may be excluded. Okay, like that also we can do. So the commercially available serum-free media are MCDB 131 for endothelial cells and LHC9 for bronchial epithelium. And serum substitutes are also there, which are commercially available. Okay, we can add that. So these are the brand names and what you call the substituents and the suppliers. Okay, sorry. So the nutrients which are present in the serum free media are iron, copper, and many minerals. So they are present in the serum free media. And some rare minerals are also required, which is still lacking. So selenium around 20 nanomolar can be added, okay, which is in the formulas. And there appears to be some requirement of lipids or lipid precursors such as choline, linoleic acid, then ethanolamine, phosphoethanolamine, which are all the components of the lipid membrane lipids, right? Phospholipids which are present in the cell membrane, plasma membrane, so they can be individually added. So other than that, we have hormones. So even hormones can be used to replace serum, which include growth factor, growth hormone, which is somatotropin, insulin, then hydrocortisone, then triiodothyronine. So here hydrocortisone improves the cloning efficiency of glial and fibroblasts, and also for maintenance of epidermal keratinocytes. Then T3 is necessary for supplement of dog kidney cells, which is MDCK. And other combination like estrogen, androgen, progesterone can be combined with hydrocortisone or prolactin, and we can use for mammary epithelium. Then other proteins and polyamides. The inclusion in media of proteins such as bovine serum albumin can be done, and the tissue extracts from a bovine, okay, bovine pituitary extract can be used in combination with keratinocyte serum free media. And this BSA, bovine serum albumin, which is fatty acid free, can be used at 1 to 10 milligram per ml concentration. Then transferrin protein can be used, which will carry the iron. Then recombinant transferrin is also nowadays available. So it is nothing but, so overall, after discussing all these things, so all these components are present in the serum naturally. So we can directly use the serum, but we don't know what is the concentration. But if you don't want serum, you don't want to take any risk, then you can choose each one. So this requires a great amount of research and great amount of understanding of what type of cell lines you are doing. So you should study about that cells. So what is required for growth of cells. So then you have to choose each and every component carefully, and concentrations also. You have to standardize. Then you can add them individually to improve the growth. So all are commercially available nowadays. So we can just procure and we can use the concentration. Then next is viscosity. So one of the actions of serum is to increase the viscosity of the media. So usually in serum containing media will be more viscous. So this is important in stirred suspension cultures. So if when it is more viscous, the shear stress will be minimized, because in suspension cultures there will be stirring, then it will generate some kind of stress, so that can be minimized. And carboxymethyl cellulose, polyvinyl pyrrolidine, and non-ionic surfactant, which is pluronic F68. So they all can be added to minimize the mechanical damage when you are not using the serum. So then we have how to select the serum free media. So serum free media is to promote the selective growth of a particular type of cell. So we have to choose, we have n number of serum free medias. So you have to select, like for MCDB 153 for epidermal keratinocytes.
Good afternoon, ma'am.
Yes.
Excuse me, ma'am.
Any
Please
question?
expand the screen. Possible.
Your voice is breaking.
Please full screen.
Full screen. Okay.
Thank
Yeah.
you.
Okay. So we have to choose each type of like media for each type of cell culture. So these are the various types of media which are commercially available, and we have commercial suppliers, and we have the list of cell lines. So before conducting an experiment, we can choose which type of cell requires what kind of media, what are the requirements. You have to do some kind of research and literature. Okay, then you have to choose, so to save time, money, everything, right? So this is about, sorry, yeah, this is about the requirements of, almost we have discussed all the components of cell culture media. Okay, so how to maintain also we have discussed in the brief. Now we will discuss about the stem cells, the next topic. Okay, again we will come to these topics, so which will be repetitive in maybe in the next classes, but I will just then I will tell you in brief. Okay, because already we have discussed in detail. So if you have any doubts, you can just ask at the end of the class. So meanwhile, we will discuss about the stem cells. Okay, I will share the slides. Yeah. So stem cells, it is of great demand now. Like almost every research is focused on the stem cells. So stem cells are the one of the most fascinating areas of biology today, because it has lot of potential, and there is a minimum amount of information available. It means it is not the study, but how to bring that into application. How can we use stem cells in treatments? So that is our main objective. So stem cells are a special kind of cells that have ability to divide indefinitely and have the potential to give rise to specialized cells. So that means whenever we are in embryonic stage, like after fertilization, a single cell is formed, which is called zygote. So zygote, the basic question of developmental biology is, zygote is a single cell from where we have got this body, complete different types of cell types, different number of cells were developed from the zygote. So how this single cell is giving rise to these many different types of cells? Okay, now we have zygote is not a specialized cell, means it can perform any action or it can develop into any type of cell. But after we are born, like before also, like we will specialized cells are formed. So what are specialized cell? That means a cell which is committed to perform a single type of action. Like you have neurons which will perform only something related to brain. It is a brain cell. Then kidney cells, liver cells, lung cells, like that. We have different types of specialized cells. But when we are in embryonic state, the cells are not specialized. They can perform any type of action, and they can differentiate into any type of cell. So that set of cells which are capable of becoming any type of cell, so they are called as stem cells. So they are blank cells in simple terms, like they are unspecialized, they can become anything. Okay, they are capable of dividing and renewing themselves for longer period of time. So when they become specialized, we will have different types of mechanisms which to stop the cell growth. But the stem cells, they have the capability of dividing and renewing, because they are required for not only the growth of the body, but also to replace the old cells. Now, if we see blood cells, after 120 days, the old cells will be gone and new cells will be replaced. The old cells are replaced with the new cells. So from where the new cells are coming? From bone marrow, we have some stem cells which will become the specialized blood cells. So this happens throughout our lifetime. So they have to continuously divide. And also skin cells. Okay, like after dead skin is replaced by a fresh skin. So that means it is coming from where? Like from stem cells. They will be continuously dividing throughout our lifetime, and they have the potential to give rise to specialized cell types through differentiation. And plasticity is nothing but the flexibility. They can adapt to the environment. So what are the unique properties of all stem cells? The stem cells are unspecialized. Okay, the fundamental property of stem cell is they don't form any tissue specific structure, the morphology. Already when we discussed the general histology, we have seen different morphologies, different structures, functions of different tissues, but that doesn't apply to stem cells, and they don't perform any tissue specific functions. Okay, and proliferation, they keep on dividing and renewing themselves for indefinite periods. Then differentiation, they can give rise to specialized tissue. So under certain conditions, physiological and experimental conditions, these unspecialized cells will become specialized cell. So in physiological conditions, normal, like I told you, like to replace the old cells with the new cells, that is physiological. So that will be a continuous process. Whereas experimental conditions means, suppose in lab, if you want to study something, okay, you don't have that, like if you see um, like brain cells, neurons, they don't divide. So if you want to study, you can just select some stem cells and you can add some chemicals which will make them to differentiate into neurons. So you can study what are the mechanisms which are involved in the differentiation. Like that we can do some experiments. Okay, then plasticity, stem cell from one tissue may be able to give rise to cell types of different tissue. So this is nothing but the flexibility or plasticity. So here the blood cells can become neurons, liver cells producing insulin, like that. Okay. So this is the timeline of the stem cell. So single cell embryo is a totipotent. So what is totipotency? That has the ability to give rise to complete organism. So that is what is happening when a zygote is formed, from where a complete organism is developed. So that is totipotent. But when it becomes 5 to 7 day embryo, the embryonic stem cells, they are pluripotent, means they cannot give rise to the complete organism, but they will give rise to different types of cells. Okay, then cord blood stem cells or placental stem cells. So nowadays, like there is an increased awareness regarding this cord blood stem cells, and people are actually saving the cord blood or stem cells in the stem cell banks, so that stem cells can be used for the baby in future. Okay, so they are multipotent, means many type of cells can be generated from this cord blood or placental stem cells. Then infant and adult stem cells. So even small babies and even we as an adults can get the stem cells, but they are multipotent. That means we can get the different types of stem cells, but not all types. Okay, we have some limitation. So totipotent means, toti means whole. So it can give rise to any type of stem cell and complete organism can be formed. Then pluri means many, from inner cell mass. So when the embryo is formed, it will form a ball like cells which is called blastocyst. So from blastocyst, we have in the during blastula stage or blastocyst stage, the outer cells, outer layer of the cells form the trophoblast, and inner we have a mass of cells called inner cell mass, which will give embryonic stem cells, which can give many types of specialized cells. Multipotent means several. It is not many, but we can get some type of specialized cells. So that is the difference here. So the fertilized egg will be totipotent. So this can form embryo and placenta. So we'll get the totipotent stem cells. Then it will become blastocyst containing pluripotent stem cells. So in the blastocyst, you can see the red nucleus containing cells which are surrounding, forming an outer layer, which are called trophoblasts. And inside we have some inner cell mass. That cells are pluripotent. So then from this pluripotent stem cells, we will get hematopoietic stem cells, neural stem cells, mesenchymal stem cells, which are multipotent. So these hematopoietic stem cells can give rise to different types of blood cells. Then neural stem cells can give rise to cells of nervous system, like tissue specific they are. Okay. Once they become the differentiated cell, they are now fully mature. They cannot be reversed. So nowadays, even that reversal is also, the research is going on that. So we will look into the terminology. So totipotent stems means these cells have the potential to become any type of cell in the adult body, any type in the adult body, any cell of the extraembryonic membrane, like placenta, then amnion. These are the extraembryonic membranes. And the only totipotent cells are the fertilized egg, and the first 4 or so cells produced by its cleavage. So only one cell, that is zygote, that becomes 2, 2 to 4. Only till that time they are totipotent. Once they become 4 cells or so, they will become pluripotent. Okay. And pluripotent stem cells, they are true stem cells which have the potential to make any differentiated cell in the body, but they cannot give rise to placenta, which is derived from the trophoblast. So here the same thing, like this in vivo means within the body, the fertilized egg, which is totipotent, becomes 8 cell embryo. Then this is blastocyst. Then this become pluripotent, which can give rise to different types of differentiated cells. So then 2 types of pluripotent stem cells are found. They are embryonic stem cells, embryonic germ cells. So what are embryonic stem cells? So they are isolated from the inner cell mass of the blastocyst, the stage of embryonic development when the implantation occurs. Okay, so what happens after fertilization? A single, I told you, like zygote is formed. Zygote starts dividing. It will become 2, 4, 8, 16, like that. Once it reaches some number of cells, it will form a ball-like structure, which is called blastocyst. So here it will form a single layer of cells, which is called trophoblast, and inside we'll have the inner cell mass. Now this ball goes and attaches to the endometrium layer of the uterus. So that process is called implantation. Once it goes and binds to the endometrium, then it starts growing. It will start rearranging the cells, which is called gastrulation. Then after gastrulation, 3 layers like ectoderm, endoderm, mesoderm are formed. From these germ layers, we will get different organs like skin, liver, lungs, gut, all these things. And once the organs are formed, it will form a structure of organism. Okay, so all this happens. So for that, the whatever the cells inner which are present in the inner cell mass of blastocyst, they are called as embryonic stem cells. So for humans, excess embryos produced during in vitro fertilization, they produce this embryonic stem cells. So what you might get the doubt, like from where we will get the embryonic stem cells? Because like usually we cannot isolate. It is we cannot even see during embryonic stage. So from where we get all these things? So whenever the couples they perform in vitro fertilization, usually they don't go with the single embryo, because to increase the rate of the success, 3-4 embryos will be developed and they will insert back into the mother's womb. So here the some may not be, like all may not get successful, some may be aborted. Okay, so, from such type of excess embryos which are produced, and everybody may not be able to if all are successful, if they have 3 or 4 babies, they may not have that capacity to maintain all of them. So at the embryonic stage only they may decide, like they need only 1 embryo or 2 embryos, like that. If there are any excess embryos, from where we can get this embryonic stem cells? They are the source of stem cells. The next is embryonic germ cells. They are derived from the part of human embryo or fetus, which ultimately produce eggs or sperms. So during fetal stage only, the germ cells will be formed. In future, what are the cells which produces eggs and or sperm based on the gender? And even by birth, female babies, they will have the number of eggs which are required for the whole lifespan. Okay, they will be in inactive stage. Once they attain puberty, they will get activated. Okay, so like that we have 2 types of pluripotent stem cells. So these types of pluripotent stem cells can be used only be isolated from embryonic or fetal tissue, and can be grown in culture, but only with special methods to prevent them from differentiation. So that means we have to maintain them in stem cell condition. If once they get differentiated, we cannot use them. So here, unused frozen embryos which are can be used as a source of stem cells. So like from these pluripotent stem cells, pancreatic cells, islet cells or blood cells, heart cells, like that we can generate the differentiated cells. So tens of thousands of frozen embryos are routinely destroyed when couples finish their treatment. So once they finish the treatment, many embryos, excess embryos will be left over. So these embryos can be used to produce stem cells. They can be used for doing research on regenerative medicine. Okay, so we produce any type of tissue using these stem cells. So like that, to treat any illness, like that we can do. So here, what kind of techniques are there? Sorry, yeah. So here it is human therapeutic cloning. So how can we perform that? Here from patient, the biopsy, through biopsy, the cells are removed, like from cheek cells or bone cells. And we can insert this cell nucleus into the oocyte or egg, from where the nucleus is removed. So we are cloning, simply like how the doll is generated. So in the same way, we are taking the somatic cell nuclei and we are inserting in the enucleated egg. And this, by the somatic cell nuclear transfer, we are getting the somatic cell nucleus into the egg whose nucleus is removed, from where we get the pluripotent stem cells. Okay, from these pluripotent stem cells we can make them to produce pancreatic cells, hematopoietic cells, cardiomyocytes, like heart cells, and we can use them back to the patient. So this is nothing but a somatic cell nuclear transfer, from where the nucleus of a donated egg is removed and replaced with the nucleus of a mature somatic cell, maybe skin cell or any other cell. and no sperm is involved in this process, and no embryo is created in the woman's womb. You're only generating the stem cells, okay, which will be further developed into specialized cells so that we can use that for the treatment. So how are these embryonic stem cells harvested? So growing cells in the laboratory. So why we are discussing all this? Because we have already discussed the cell culture. So in that cell culture, not only the differentiated cell types, but the stem cells also can be cultured. So human embryonic stem cells, which are derived from 4 to 5 day old blastocyst, can be cultured. So blastocyst, I told you the trophoblast, which is the outer layer of cells that surround the blastocyst. From this trophoblast, we will get the placenta. Then we have a small portion of cells called inner cell mass, and inside this blastocyst, we have the cavity, hollow cavity, which is called blastocele. So this forms the body cavity. Okay, when this embryo becomes a matured, like a newborn, so that will become the body cavity. And the inner cell mass, it have maybe approximately 30 cells. They will rearrange themselves to form 3 germ layers, which are ectoderm, endoderm, and mesoderm. So here, this is the embryogenesis. Like first in the day 1, it is the fertilized egg. On day 2, it will become 2-cell embryo. On 3, 4, it will become multi-cell embryo. And 5 to 6, it will, on day 5 or 6, it will become the blastocyst. By 11th or 14th day, it will become the differentiated tissue by this blastocyst. So here, blastocyst can be, we can isolate the blastocyst from the in vitro fertilization clinics. Okay, so a blastocyst is a hollow ball of cells with a small clump of stem cells which are present inside. So here you have the stem cells, and inside you have the blueprint cells. So which will give you the like a blueprint of like which organ has to be formed, like that. So to remove the stem cells, a blastocyst is opened and the stem cells are removed from the pipette using the pipette. And then the stem cells which are removed using pipette are placed on a dish, and they have to be given the media, required media. And each blastocyst will become one stem cell line. And so in this petri dish, we have, so here in the petri dish, we have different types of, means different number of stem cells. So now we have to give growth factors and chemicals so that it will become different differentiated cells like pancreatic cells or neurons or muscle cells. Okay, so I think you have understood. I will just check once again. So from in vitro fertilization clinics, whatever the leftover embryos are present, so from that embryos, usually we will isolate the stem cells, that is inner cell mass cells, using the pipette. Then that cells which are removed using pipette should be placed in a petri dish. And suppose if we give some type of chemical, it will become pancreatic islet. Or if you are giving another type of chemical, it will become the neuron. Or any other growth factor, it will become muscle. So we will have some protocols. So based on what cell type you want, so we have to supply that chemicals or molecules to stem cells so that they will be differentiated into the specific cell type. So what are the cell culture techniques? So first thing is isolate and transfer the inner cell mass into plastic culture dish which contain the medium. And now the cells will divide and spread over the petri dish. And the inner surface of the culture dish is coated with embryonic mouse embryonic skin cells that have been treated so that they will not divide. So what are these mouse embryonic skin cells? They are like feeder layers. That means in the culture media, in the culture dish, petri dish, you have media, and above that you have mouse embryonic skin cells which won't divide, and above that you are adding this stem cells, so that these mouse skin cells, they will supply the growth factors required for the growth of the inner cell mass. So this coating is called feeder layer. So the feeder cells, they provide embryonic stem cells with a sticky surface for attachment, and also they release nutrients. And recently what the scientists have discovered are, there are some methods, sorry, for growing embryonic stem cells without mouse feeder stem cells. So that why we need to eliminate the mouse feeder cells? Because to eliminate infection by viruses or any other mouse molecules. So because they are of different origin, suppose what for this first thing we have to remember is, what is the purpose? It is only for simple research, if you, or are we applying this in the real treatment? Suppose if you want to use it for real treatment, you have to directly use the stem cells into another human being. So if that cells are contaminated with the viruses which are there in the mouse feeder layer, then that cells cannot be used for the treatment, because the person will get affected with some other virus or some other viral disease. Therefore, in real time, when we are doing for application purpose, it's better to avoid the feeder layers. And the embryonic stem cells are removed gently and plated into several culture dishes. Once the embryonic stem cells are grown, you can remove them and place them in different, maybe 5 or 10, like that, based on the requirement. You have to place them in the different petri dishes. Then after several days, they proliferate and they form crowd in a culture dish. Now they have to be removed, like subculturing must be done. So they have to be removed and placed in the fresh culture meat dish. And the process of plating the cells in repeated several times for several months, then this is called subculturing. And each cycle of subculturing is called as passage. That means suppose in from one dish, I am splitting that into 2 dishes. That is first passage. And from 2, I'm making them into 4. That is second passage. Like after each cycle of subculturing, that is called as passage. After 6 months or more, the inner cell mass yield millions of embryonic stem cells. Now they are pluripotent, and they appear genetically normal, and now they are referred as embryonic stem cell lines. Okay. So this is the stem cell cultivation, the same steps. From in vitro fertilization egg, we are getting blastocyst stage. From there, inner cell mass is removed in the petri dish, and from there we are using some chemicals to differentiate them. Then multipotent stem cells. So these are the true stem cells, but cannot differentiate for very longer period of time. They can differentiate only for limited number of types. Okay, for example, bone marrow cells contain multipotent stem cells which can give only blood cells, but not other types of cells. Right? So these multipotent stem cells are found in adult animals, including human beings. So like whatever the replacements which will be done in like a general turnover process, all these are nothing but the multipotent stem cells, like brain, liver. So they contain to replace the dead or damaged cells. These adult stem cells, when accumulate sufficient mutations, they will become cancerous cells. Okay, so in stem cells, if any mutation happens, that will be more dangerous, because that will be continuously dividing. So if normal, after differentiated cells, if there is any mutation, we can remove that part. But if that happens in the stem cells, so every time when the stem cell will get differentiated, that will become a cancerous cell. So that is more dangerous. Like we have brain cell, heart cell, bone marrow. So from brain cell, we are getting neural cells. Then from heart, we are getting cardiac muscle. And from bone marrow, we get blood cells. Then adult stem cells. An adult stem cell, also called as somatic stem cell, which are undifferentiated cells and can form, we can see them in between differentiated cells only, and they can renew themselves and differentiate to give major specialized type of the tissue or organ. So the primary role is to maintain and repair the tissue. That's it. It is not de-differentiating or differentiate into some other type, not like that. Only to maintain and repair that particular tissue. Okay, their origin is unknown, but for some cells we can see hematopoietic stem cells can form all types of blood cells. Then stromal cells can generate cartilage, fat and fibrous connective tissue. And brain stem cells can form astrocytes, oligodendrocytes, neurons, etc. So this is how the different cell types are formed. So this is for brain cells. Okay, from brain cells we are getting CNS stem cells, central nervous system stem cells. Like that we have different types. Please mute yourself. So contrary to embryonic stem cells, which are pluripotent, adult stem cells are more restricted. Okay, they cannot give rise to many types of cells also, but they are very much specific to a lineage. Only that particular type of cells will be formed only for maintenance and repair of that particular tissue. So these adult stem cells we see in bone marrow and other tissues, and stem cells which are outside the bone marrow are called as tissue stem cells. In the tissue stem cells, okay, they are located in the sites called niches. Okay, only at particular sites they are present. So you can see here stem cell niches in various tissues. So we have in the hair follicle, then crypts of colon. So in the colon we can see this blue color. Then canals of herring, then limbus of cornea. So like that only in some portions we can see. So what are the, when we compare the embryonic versus adult stem cells, so what are the differences? So embryonic stem cells are totipotent. They can form any type of cell type. And we know the source of stem cells from embryo, and that is inner cell mass. And large numbers can be harvested from embryos, and they may cause immune rejection. So rejection of embryonic stem cells by recipient has not been shown yet, but there is a chance. Then adult stem cells, they are multi or pluripotent, can differentiate into only some types of cells, and the outcome is also limited. The source is unknown, and limited number can be obtained because they are adult already, they are differentiated mostly, and we will have only limited number of cells. It is more difficult to isolate because they are present only in some pockets, so it is very difficult to isolate, and less likely to cause immune rejection because already the MHC molecules all will be developed. So we will have a clear like we will have the clarity. So if we match, if the MHC is matched, so it will be definitely, the rejection rate will be reduced since the patient's own cells can be used. And now the most important is cord blood. So umbilical cord blood is also known as placental blood. So from umbilical cord, we will remove the blood. Okay, so it is a blood that flows in the circulation of the developing fetus in the womb. After the baby's birth, so whatever the leftover blood which is present in the umbilical cord and placenta, that blood is called as cord blood, and it is a rich source of stem cells. So we can isolate the stem cells from this umbilical cord. So now there's a big demand for this. The umbilical cord blood is a rich source of primitive stem cells compared to adult stem cells. So because once we become adults, so that the chance of differentiation is reduced, only some types of stem cells only we can get. But from primitive stem cells we can get lot many types of stem cells. Therefore they can expand rapidly, and stored cord blood stem cells from a child is perfect match for that child, right? So it's very good to store these umbilical stem cells, and this allows for autologous transplant if needed, and no risk of graft versus host disease. That means there is no risk of rejection, only if it will 100% match with the child, because when the child become adult we can use these stem cells. So the cord blood stem cells are a close match for siblings or family members also, with low risk of graft versus host disease. So making decision to collect baby's cord blood cells, yes, if there is a family history of malignant, benign or inherited disorders. So it's very important to store, it is always good if we store the cord blood cells normally also, but if there is a family history of any cancer, whether it is malignant or benign or any other disease or any genetic disorders, it is very good so to store the cord blood stem cells of the baby, because for the treatment it will be very useful. And even if there is any absence of health risk factors, so it's very good to store the cord blood cells, because they have a lot of benefits in the future, and if the cost is affordable, and so it is very good to, it is a value, it is nothing but giving a second life to the baby. So it's always yes. So it is only one chance to collect. So once you, if the baby is born, then if you go try also we cannot collect. So it is the quick decision that we have to take. Okay. So how this cord blood stem cells are collected? So this blood is collected by the physician after the baby is born. The cord is cut. So once the baby is delivered, so usually cord clamping will be done. That means the clips will be placed and they will cut the placenta. Okay, they will separate the placenta from the baby. Okay, they will cut the cord. So it takes less than 5 minutes. There is no pain. So from that cord they will collect the blood. So there is no pain for the mother or newborn, and no harm or no risk. It is a waste thing, they will just throw it off even if we don't collect also. And even the placenta, whatever the placenta which is delivered, even that can be stored if required. And this cord blood containing stem cells are sent to the cord blood bank, either private or public, where that can be processed and they will be stored. So there are two types of blood blood banks. We have cord blood banks, so private and public. Public means they will be stored and that can be used to anyone. Okay, so we have to give the permission prior, and wherever this if this matches with some other person, so they can use. But private means, even in the private we have what you call individual and community based. So community means whatever the stem cells which are matching some 80-90%, so all these will be stored as a community. That means suppose if my baby stem cells and other baby stem cells are also matching to 80 or 90%, so they can use the stem cells for each other. So now the chance of stem cells, the availability of stem cells will become more, right? So that is community. So we can choose for private or public, community or individual, and we can store that. So once the cord blades are collected, they will be stored in the liquid nitrogen at minus 196 degrees. So this can be stored till 100 years almost. Okay, So immediately after the baby is delivered, umbilical cord is clamped. And after delivery of placenta, the placenta is placed on supporting frame and the cord is cleaned and a needle is inserted into the umbilical vein, and from there the cord blood is collected, and it is a closed system and the blood is drained by standard gravity phlebotomy. Okay, so here there will be no pain or nothing for that. And the second method, which involves collecting the cord blood, is from the placenta, which is still in the mother's womb. Okay, this has two advantage.
What is
One,
meant by phlebotomy?
it is like a separation technique, removal of blood. Okay, by standard method of removing the blood.
Thank you.
Okay. So the second method is while the placenta is already there in the womb. Okay. So here the advantages which are associated is collection begins earlier before the blood is, there is a chance of clotting. So once it is separated from the body, so there is a chance of clotting. So before they clot, we can collect the blood. And it uses the contraction of the uterus to enhance the blood drainage. So it is nothing but, so during contraction, the blood pressure will be increased. Okay, so the blood drain will be more. So you can collect large amount of blood from that. Okay, so this is the advantage, but disadvantage is, so it will interfere with the delivery. Okay, so it's always better to collect after the placenta is delivered. Okay, usually we get 60 to 120 ml of blood. Okay, that will be collected. So here the umbilical stem cells, sorry. So they are nothing but the adult stem cells which are of infant origin and greater compatibility and less expensive. And the three important functions are plasticity, they can change into any type of cell lines. Then homing, so that means they can travel to the site of tissue damage. And engraftment, that means to unite with the other tissues, so they can work with along the other tissues. Okay, so what are the potential uses of stem cells? So the basic research is done. So the basic research is nothing but the clarification of complex events that are occurring during human development and molecular basis of cancer. So that's what, to study the developmental biology. Okay, by basic research, like we don't know, like from stem cell to now whatever we have discussed the concepts, so how we know like what is happening, it is through some basic research that was done. And still many more concepts are there which are not clear, so all that can be studied from the stem cells. And how the cancer, like what mutations or what mechanisms are involved in the cancer generation of cancer, so that can be studied. And molecular mechanism for gene control, so how the genes are controlled, that can be studied. And signals which are involved in gene expression and differentiation of the stem cells. And stem cell theory of cancer, how the cancer stem cells are generated, all these can be studied. And the uses are, so how it is used in the biotechnology means, so for drug discovery and development, so stem cells can provide specific cell types to test new drugs. So suppose I want to test some drug in some particular type of tissue, so no need of removing tissue or something, we can just use this stem cells to differentiate into that particular tissue, and I can do the research. So the safety testing of new drugs on differentiated cell lines and screening of potential drugs. So cancer cell lines are already being used to screen the anti-cancer drugs. Now, if we have the pluripotent stem cells, so this will give a wide range of drug testing on different types of cells, and it will reduce the animal testing. No need to kill different animals to isolate the tissues. So first, even if we want to do the cancer research, first we have to like inject cancer into the animal, and then we have to kill, so we have to make it suffer. So in order to prevent all those things, it's always good to go for the stem cells. And cell based theories, so we have regenerative theory, like we have some diseases which cannot be cured, like Alzheimer's, Parkinson's, ALS, spinal cord injury, strokes. So that can be reversed if there is any success in this cell based therapies. Then tissue repair, so to regenerate spinal cord, heart tissue, or any major tissue in the body. Then heart disease, so adult bone marrow stem cells can be injected into the arteries of heart, okay, so that it will improve the cardiac function in patients with heart attacks. And replace skin, so during burns, like that, we can replace the skin by using our own stem cells instead of removing the skin from any other part, so where the like chances of infection all are more. Okay. Then rheumatoid arthritis, so which is also a genetic disease. Okay, so adult stem cells may be helpful in jump starting the repair of eroded cartilage. Then type 1 diabetes, so pancreatic cells which do not produce insulin, so we can replace them with the embryonic stem cells, okay, so that we can train them to secrete insulin. So basic research focused on understanding how embryonic stem cells might be trained to become pancreatic islets to need to secrete insulin. Okay, so that by using this we can increase the production of insulin. And the other uses are gene therapy, so where we can manipulate the genes and we can insert back into the body. And if scientists can figure out how stem cells choose among their career options, they might be able to direct them to become a specific cell type and repair a specific tissue. That means now from each stem cell is becoming only one type of cell finally. Though it has the capability to becoming any type of cell, but it won't become any type. It will become either brain cell or liver cell or lung cell. Only one type of differentiation happens. So how this is happening, if we know that process, so we can easily make them to differentiate and we can use them for specific treatments. And therapies that might work, okay, are if we can remove the bone marrow and the stem cells are isolated and we can culture them, and these cells are treated with a modified virus with a gene. So now this gene gets inserted into the stem cell, and whenever the stem cell, so the virus is taken up by the individual cell and the therapeutic gene goes into the cell's nuclei. So this is the corrected gene, the proper gene. Now this can be injected into the bloodstream and they will go, and these treated cells will respond to the injury signals and they will try to regenerate the muscle or any other tissue. Okay, and it will treat the damage. Okay, so this is a type of therapy which might work. So some are only still in theoretical levels, they are in research level, they are not into application. So how this hematopoietic stem cell transplantation is done, so this is for steady adult stem cells, and this can give rise to all types of blood cells, and this is very good, it is well established for clinical use. So any procedure where hematopoietic stem cells of any donor or any source is given to the recipient by replacing the hematopoietic system, so that is nothing but the hematopoietic stem cell transplantation. Okay, so that means we are replacing the damaged cells with the hematopoietic system from the donor. Okay, so he is a person like Donald Thomas. He got Nobel Prize in 1990 for first successful hematopoietic stem cell transplantation for the treatment of acute leukemias, cancers. So he used this method. So what are these hematopoietic stem cells? They are defined as the cells with the ability to achieve long-term reconstruction of both myeloid and lymphoid lineage. So in this we have two types of lineage, myeloid and lymphoid. So they can give both. So the features of these hematopoietic stem cells are they have regenerative capacity, they have the ability to home marrow spaces following IV injection, so intravenous injection, and they can be cryopreserved. So here you can see from the hematopoietic stem cells, so myeloid progenitor below and lymphoid progenitor above. From where we get, from lymphoid we get T cells, NK cells, dendritic cells. Then from myeloid precursor cells we get granulocytes, macrophages, platelets, red cells, red blood cells. So all this we can get. So here we have to look for the markers. We have CD34, CD135, like that. So they are the determinants. CD means, okay, so that it is a marker. So each type will have some marker. So by marker you can select what is that cell type. Suppose if you have regulatory T cells, you will have CD4, CD25 and FOXP3. So this is, if these three are present, then you can say that is a regulatory T cells. Like that each cell will have a specific marker. So what is the basic principle here is to replenish bone marrow cells to eradicate any disease or chemotherapy or radiation in cancers, especially in cancers. So the same thing, if the stem cells can be removed from bone marrow or peripheral blood or umbilical cord blood. So this is how they remove from the bone marrow. So marrow is obtained by multiple aspirations from the posterior iliac crest. So from back side, by giving anesthesia, they will remove some bone marrow. And if we are removing from sternum, we can get large quantities, and puncture has to be done. And the target volume is 10 to 15 mg per kg of recipient's weight. Based on how many kgs of weight the donor is there, so based on that we can get 10 to 15 mg per kg. And the marrow is collected in a heparinized syringe, and they remove fat and bony spicule, everything is removed, and they are further processed. And removal of RBCs to prevent hemolysis in ABO incompatible transplants, and removal of immunocompetent donor T cells to prevent graft versus host disease, and remove possible contamination of tumor cells. Then this marrow is transfused immediately after harvesting, and they can be delayed up to 24 hours to see if there is any adverse consequences. Then we can collect the stem cells from the bone marrow. Then peripheral blood. So peripheral bloods from where we get the stem cells are, these cells are now the most common source of stem cells. And here peripheral blood stem cells are collected from leukapheresis after the donor has treated with the hematopoietic growth factors. That means nothing but we have to give the growth factors. Okay, just a second. So once we give the growth factors, or if he or she is already under chemotherapy or growth factors are given, then we have to isolate. And for this the malignancy, okay, malignancy cyclophosphamide based chemotherapy or colony stimulating factors can be used for this purpose. Okay, granulocyte colony stimulating factors, so they can be used for before isolating the peripheral blood stem cells. Okay, then the donors are treated with the growth factor for 4 to 6 days, following which the stem cells are collected in 1 to 2 4-hour session. So like that they will collect. And in autologous setting transplantation, so we have to select the CD34 plus cells per kg. So that means I told you CD here is a marker, CD34 plus. So that means the cells which are having the CD34 marker, that has to be isolated. So how we isolate, that means it is by flow cytometry. So it is FACS actually, flow activated fluorescence activated stem cells, cell separation, sorry. So by using this FACS, that is fluorescence activated cell sorter, we can actually sort the cells by applying some fluorescence. That means suppose you have CD34, you want only CD34 plus cells, but you have collected the whole blood and you are adding a antibody which is specific to CD34, and you are just tagging a fluorescence tag, either FITC or P, like it will emit green light or red light, like that. Now you have to add that reagent into this sample so that this antibody goes and binds to the CD34. Okay, when we are sorting, there is a machine which is called FACS, and that will sort the cells which are only having the fluorescence. Like that, we can get only the cells which are having the green fluorescence. That means they are having the CD34 specific antibodies. When they are tagged, means already they are cells which are displaying CD34 markers. So like that we can separate. Okay, and here this is apheresis. Apheresis is nothing but a machine or a device which receives the blood from the donor, and it will separate them into various components like platelets, plasma, red blood cells, white blood cells, like that. Okay, so this is, sorry, yeah, peripheral blood stem cell transplant. So here we can collect them from the bone marrow stem cells and extracted from within the bones. Okay, and this is very invasive treatment, and we can easily get the stem cells from here. This is cord blood collection from the umbilical cord. When it is cut, usually, so I told you the mechanism, like to puncture, actually to puncture and to remove the blood. Then storage. So bone marrow cells can be cryopreserved for prolonged time, and it is necessary for autologous hematopoietic stem cells because the cells must be harvested months in advance to the transplant treatment. So before the treatment, before months only, that has to be harvested back, and that should be differentiated if required, and then it should be transplanted, because the HLA all should match. Okay, and in allogenic transplants, fresh hematopoietic stem cells are preferred in order to avoid any cell loss which can happen during freezing and thawing process. And the graft undergoes HLA typing and cell counts and testing for virus. Is there any virus or any what is the HLA type? Okay, that has to match between donor and recipient. How many number of cells, all this they will check. And allogenic cord blood is stored frozen at the cord blood bank, and that is which is obtained during the time of birth. And the cryopreserved hematopoietic stem cells. So usually I told you, like in the last class, before cryopreservation we add cryopreservants like DMSO should be added, so that it will prevent them from any cell damage. And slowly we have to thaw. Directly you cannot keep them directly from -196 to room temperature, so it will damage the cells. And it can be stored for many years, like 100 years, using cryo freezer. So here the cellular characteristics of various sources of stem cells. So the stem cell content, it is adequate in bone marrow, it is good in peripheral blood, and low in cord blood. So like that, progenitor cell content also adequate in bone marrow. And T cell count, it is all high in peripheral blood, and risk of tumor cell contamination is high in bone marrow. So like that we have to select based on that. And these are the clinical characteristics. So HLA matching, so close matching is required for peripheral blood, but in cord blood it will be same. Okay, then enfranchisement or risk of acute graft versus host disease, it is fastest in peripheral blood, and risk of chronic graft versus host disease is highest in peripheral blood. Then indication of then hematopoietic cells, where we have to use this is in neoplastic disorders like hematological malignancies, multiple myeloma, lymphoma, leukemia, solid tumors, or non-neoplastic disorders like autoimmune disease, inborn error of metabolism, where we can use this hematopoietic stem cells. So like this. So yeah, ethics. So how far it is ethical? So even there is a lot of debate is going on this. So ethical issues concerning the use of stem cells are, so directly I told you, like by removal from the embryo, that is more it is concerned of ethical. So origin and method of stem cell production, and small number of adult stem cells are found in the human body, but the best source is embryo or fetus. So but fetal stem cells, how we have to isolate? So in some women, like in some patients, they are aborting the fetus only for the money, and from that aborted fetus the fetal stem cells are generated. So if it happens spontaneously, that's fine, but some may so how we are collecting, some are like subjected to the ethical concerns. Okay. And so the key ethical issues are like, does it count as a person? Does the embryo count as a person? And there are some religious beliefs which are associated. Okay, so all these, so the public policies, what are the demands, so all these are the ethical concerns. So yeah, are we playing with the nature or like that? So this is all about the stem cells. So if you have any quick questions, you can ask. Already time is up. If you have any one or two questions. Yes, no questions? Is it clear?
Yes, ma'am.
Okay. So I don't have this group, I think. I'm not there in the group. Okay, at the end of the class, at the last class, I will share all the PPTs. Okay, just remind me, because every day sharing it will be difficult. So at the in the last class, I will share all the PPTs. Okay. Okay, then fine.
Okay, ma'am.
Yeah, thank you. We will discuss in the next class.